Okay, hi guys. In this short video, we're just going to look at how you can go and model different epigenetic concepts with students who are doing A-level biology. But epigenetics is one of the more abstract concepts that they'll come across. It's one of the more difficult parts of the course. So helping them visualize what's actually going on within the cells using plasticine models is a really good way of how you can go and get this through to them and really help it stick in their minds. Okay, right, so I'm just going to go and put on 3 or 4 short videos just to show how different epigenetic concepts can be covered, and then we'll go and we'll just talk through a bit of the minutiae after. So RNA processing happens after the messenger RNA has been transcribed from the DNA. The DNA contains extrons and introns. Remember, only the introns go and code for genes. After the RNA has been transcribed into messenger RNA, it's then edited. Right? Sometimes this is called RNA splicing because parts of the unedited messenger RNA are spliced out. So we end up with edited messenger RNA that now only has the code for the particular protein, and at either end we go and put a cap just to go and hold it together. Transcriptional control is all to do with either promoting the transcription of messenger RNA by DNA polymerase or repressing it. Right, so this can be done in a few different ways. So repressed proteins or complexes of repressor proteins can bind onto the DNA. Right? If a repressor protein is there, that physically goes and stops the RNA polymerase from going and transcribing it. You can have promoter proteins or promoter factors that go and bind onto the DNA, which causes the RNA polymerase to bind to it more strongly, therefore you get more messenger RNA being produced. Both of those things that are like added onto it we call epigenetic changes, right? Because we're physically attaching stuff to the DNA. Right, on the DNA itself, you'll have areas called promoter regions, which are also called Tata regions because they've got the nucleotide sequence TA, TT, AA, which physically promote the transcription of the gene that comes after that region. The second type of gene control is when the DNA is wrapped around heterochromatin, right, or wrapped around this A protein complex to form heterochromatin. Right, this happens during mitosis, and it causes the chromosomes to condense. During mitosis, we don't want any of the genes to be transcribed, as the DNA could become damaged, and all of the energy that the cell's using needs to be used physically for mitosis, not to go and carry out any protein synthesis. So the DNA gets tightly wound this to form heterochromatin. The proteins will have a slight positive charge because DNA is slightly negatively charged. That's what binds it to it. So the DNA gets tightly wound around it, which means that the RNA polymerase can no longer bind to the DNA because it's not physically got enough room to move around. So therefore, no messenger RNA can be transcribed. The third type of gene control is to do with histomodification. So DNA is wrapped around histone complexes, which are made up of 8 proteins that can be modified to either make them less positive or more positive. Right? So these histone complexes are slightly positive because DNA is slightly negative, so therefore the DNA will bind to it. If acetyl groups are added to the histone complex, that makes it less positive, so therefore the DNA binds less tightly to it and can be transcribed by RNA polymerase. If, however, these histone groups are modified by adding methyl groups to it, that makes them more positive Therefore, the DNA binds more tightly, so the RNA polymerase, well, it can't get around it, so it can no longer transcribe the genes on that DNA.
But hopefully that all made sense to you, and you could see how the different modelling activities worked. As well as doing it with plasticine, it also works equally as well using sweets, so using marshmallows to represent the histone complexes and things like that, depending on what you have available. Remember that when you are going through the models with students, ask lots of questions. Get them really, really engaged in how the genes are being turned on and off, and really focus on the different structures. After you've finished modelling it yourself, one thing that I've done in the past is I've put students into different groups with different epigenetic concepts to go and model themselves. They've gone and put together their own models, and they've presented back to each other, which really helps them and has really helped with their understanding. Right, if you've got any questions or comments, drop them down below and I will attempt to get back to you. Right, thanks guys.